And thank you all. And it's especially a pleasure when you know that students might want to be interested in your research, because that makes it extra special to talk about it. So, as Stella mentioned, we do a number of different things in my group, and so in putting my talk together, I grabbed 3 stories out of about 7 or 8 that we have going on right now, and I hope I don't disappoint anybody by the 3 that I grabbed. But I think they at least capture part of my title. So I'd like to start with just kind of an introduction and this idea that biologists have historically studied organisms part by part and celebrated the modern ability to study them molecule by molecule. And really, when I was training, this is how we did biology, and particularly I'm a biochemist, how we did biochemist. We wanted to study proteins, so we purified that protein, so there isn't another protein molecule in the whole test tube. And then we said, aha, now we're going to figure out how the biology works. Well, I think there's a problem with that, is we may figure out how the protein works, but in the process, we kind of lose the biology of that, And systems biology is really kind of this idea that we need to study things as an organism. If we really want to understand biology, we have to go beyond this reductionist approach and kind of take the opposite approach. Let's start looking at the bigger picture. Let's try to look at organisms. Now, there's lots of problems with that. People took the reductionist approach because you could actually ask a question, you could come up with a hypothesis, you could test it, and you could get an answer. The problem if you're studying a system is it's very difficult to get a direct answer to your question, or to be sure that you're really answering the question you think you are. So we've seen this really kind of explosion of these omics. And what I mean by omics is, it started with genomics. We started sequencing genomes, and this idea that we're going to look at all of the genes in an organism at once. That's the genome. Well, as we know that then the genome is, and according to the central dogma, we have translated into mRNA, and so we have transcriptomics. Let's study the mRNA in a cell, the transcriptome. Or let's study the proteins, or all of the proteins in a cell is the proteome. You can also think about post-translational modifications, which really regulate protein function. Well, we have the phosphoproteome. We have all of these different omes. And now one of the later additions to this is the metabolome, or all of the metabolites in an organism. And together, if we're going to really try to understand an organism and think about systems biology, we really have to encompass kind of all aspects of this. Now that becomes very difficult to do from a experimental standpoint. But one thing I might argue is that the genome tells us what potentially an organism could do, because it tells us what genes are present. The mRNA tells us essentially what is an organism thinking about doing, right? It's preparing to do something. The proteome, this tells us what is an organism essentially set to do. It's ready to do. It's got a protein there, it's ready to do something. But it's really the metabolites. This is the area that actually tells us that something is going on, because all of these the genome, the genes, the mRNA, and the proteins, those just tell us about potential, not that something is really happening. But if you watch a metabolite change, if you see substrate move to product, that's biology. It doesn't matter what. That is the biology that's happening. And so in some ways, I think of all of the omics, metabolomics really has the biggest potential for helping us understand systems. Because if metabolites change, biology is happening. And this kind of, falls under this overall blanket of systems biology. So here's the 3 little topics that I picked. So we're going to talk about using chemical biology, it should say to study viral infection instead of and there. We'll talk about a little that project. Then I'll go into some work we're doing with hemorrhagic shock, where we're actually looking for biomarkers. And then we'll wrap up with can we come up with essentially a protein sensor assay to make our life easier in these studies. So I want to start out with norovirus or Norwalk virus. So you may have heard of this virus. It's in the paper often as the cruise ship virus, where an entire cruise ship gets infected and they essentially have to pull everybody off and sterilize the ship. It happens in nursing homes, like this article out of the Bozeman paper from a few years ago. Also happens on college campuses pretty often. And so this is a virus that's very difficult to eliminate. It's very, just washing counters with disinfectant doesn't eliminate this virus. And currently, there is no, therapeutics and there's no vaccine against this. Now, actually, a company in Bozeman, formerly Ligocyte Pharmaceuticals, which is now Takeda Biologicals, they were bought by a company from Japan. They actually have a norovirus vaccine in clinical trials right now, but it's not available yet. But so we're studying the infection process of this virus. So this is a small icosahedral virus. So it's essentially as an RNA genome, and it has a protein capsid surrounding it. So it has no lipid envelope around it. And of course, it causes these severe gastrointestinal issues, and that's one of the things that we were interested in studying is, are there other areas that are, potential targets within the biology to look at? And so we're actually using a mouse model, the murine norovirus, because when you're working with students, you don't particularly want to be working with live norovirus if you don't have to. And this is a collaboration with Michelle Hardy from MSU, or actually she just left MSU a month or so ago. Okay, so we were culturing, these are culturing cells in tissue culture to produce virus, and then we were following the infection process. And this was a nice example of how observation is just one of the key events when you're doing research. So my graduate student noticed that when she was working with these cells, so here we're looking at control cells, and we have at the same time points the control cells and the infected, and she was saying, gosh, at 4 hours after I had my virus, I can see a difference in the microscope between these cells. And I think you might actually say you can see that too. I mean, particularly by 12 hours, you can see that these 264 raw cells become activated. They start to have these processes, and you start to see a little blebbing, and you can see they look to start rounding up. Well, in the literature, it said that if you use an antibody screen looking for viral proteins, or you look for some signaling pathways being turned on, you can't tell if a cell is infected until 12 hours. So we said, oh, come on. It really looks like you can see these sooner. So we decided, well, what if we take a proteomics approach? Let's see if we look, do we see a change in the proteins being expressed at these early time points? And so what she did is essentially she ran two-dimensional gels. So these are where we take all of the soluble proteins from a cell, and we essentially separate them based on their charge, and then we separate them based on their molecular weight. And so then what we're looking, I left off the time points, but essentially we're looking at the 4 hour and the 12 hours after this. And so what we want to do then is compare essentially these images of control and infected cells and say, so each one of these little spots is a protein. And so we can make an image comparison and say, are there any differences? Have the protein increased in abundance or decreased in abundance? And we can actually do this very accurately with this technique called differential gel electrophoresis. And so what we do is we take one set of proteins and we label those with a green fluorophore. We take another set of proteins, so for example the infected cell proteins, we label those with a blue fluorophore. Then we mix those all together and we run them on the same two-dimensional gel. And so this essentially removes any bias from the way the gel was run, makes this a very consistent process. And so we can go in and actually then use a laser and scan out of the same gel. We can pick the green labeled proteins and the blue labeled proteins. And so then we can be very quantitative in saying, is a protein increasing in abundance or decreasing in abundance? And so you do these experiments at different time points, and now we're focusing on the 4 hour time point. And in the top image, we're looking at the So the little arrows show you spots that were regulated. So either they went down in intensity, shown with red, or up in intensity, shown with blue. Okay? And so the way we know what protein that is we actually go in and we take a little pipette tip, we just poke that spot, and then we actually take the little gel plug out, we add trypsin, we digest that protein into small peptides, and we can throw that into our mass spectrometer, and we can do fragmentation analysis, and we can identify the peptides that are in there, and we can map that back onto the genome. So in this case, we're using mouse cells. So then, and we can identify precisely which protein we have in each spot. And that's one of the powers of this technique. We go from a whole proteome to picking out exactly which spots are of interest. So here's at 12 hours, okay? So at 12 hours, we see actually a larger number of proteins changing, and we see actually quite a bit of proteins that are down-regulated. Now, this is where proteomics gets troublesome, is oftentimes you get these large lists of proteins. And at this point, we're actually not seeing any viral proteins. So there's no proteins encoded by the viral genome. These are all host proteins, and some are increasing in abundance, and some are decreasing in abundance. But the problem is, what does this mean? And initially, nothing really popped out at us as to what's the biology going on. So then my graduate students started comparing the two lists. And so here's our two lists from 4 hours and 12 hours post-infection, looking at regulated proteins. And she started to notice that, so all of the proteins shown in tan here, these are all associated with apoptosis, or programmed cell death. So we got this idea, wow, I wonder if apoptosis is going on. Is that a pathway that's potentially being triggered? And so here, this our proteomics, kind of our big view approach, now we have something we can actually test and look for. And so we went on that idea of, is apoptosis involved? And so just to remind you of apoptosis and the pathways involved in this programmed cell death, so signaling for apoptosis can either come actually from inside a cell and be signaled out and back in, or it can come completely from the exterior of the cell. But it essentially involves activation of, one, there's a number of signals that can essentially lead to signaling that starts at cathepsin and this protein BIB, which activates the mitochondria, where we have these procaspases, which are cysteine proteases, which start to become active. We have cytochrome C released from the mitochondria, and this leads to further activation of caspases 3, 7, and 9, which are involved then with starting this whole program where a cell essentially is going to disassemble itself in a way that leads to no immune response in the cell, this process of apoptosis. So these cysteine proteases, or caspases, are key to apoptosis, and that's how people usually follow them. So we wanted to see, are these enzymes being turned on? And the approach we wanted to take was essentially a chemical biology approach. What we're going to do is we're going to use activity-based protein profiling. So in this method, what we're going to do is we have essentially, if we think about an enzyme, that most enzymes have a nucleophile in the active site. So they have a reactive group, a side chain of some amino acid has been properly tuned to have its chemistry so that it can essentially react with its substrate when the enzyme is active and turned on. And so you can actually design chemical, small molecules that react with these nucleophiles. And so we have a reactive group that is going to react with this nucleophile and essentially form a covalent link with that enzyme. Now, there's, if you look in the Sigma catalog, it is filled with these small molecules that react with different classes of enzymes. Okay, and this is how a lot of our, enzymology and mechanistic studies of enzymes was done 50 years ago. So these are not drugs because they're not specific, but they do react only with enzymes when the enzyme is active. And there's, so we're using these classes of molecules. Our trick is we attach these to a fluorophore so that we can essentially know when that protein gets labeled because it becomes fluorescent. And we can run the protein on a gel, for example, and pull out a fluorescent band which tells us there's an enzyme that got labeled. Or we could attach biotin and essentially use the biotin-avidin affinity for each other and directly pull that protein out of a complex biological solution. And the key thing is we only react with enzymes that are active. Now, we have actually a number of different probes. And so I'm showing you probes for different classes of enzymes. And these all have a fluorophore on them. They have a linker molecule, and then they have a reactive group that helps tag that to this class of enzymes. And so these were actually synthesized by Paul Grieco, an organic chemist in our department. And so I want to focus on this caspase probe. So this reactive group, this acyl methyl ketone, is specific or fairly specific for caspases. And actually, I'll tell you, we don't think that's quite as specific as people might lead you to believe. So we wanted to take this probe and watch this viral infection process and see, can we see caspase activation? So here is essentially our same cell culture setup, where we're looking at non-infected cells and infected cells, but now we're using a fluorescence microscope instead of just a light microscope. And so what we can see is in our non-infected cells, when we add this probe, this FLICA probe, this fluorescent probe, we can see that in the infected cells, we see caspases activated at 8 hours. We can clearly see that, and then even higher at 18 hours we see activation. So this tells us that in these virally infected cells, caspases are getting activated. So then we used a whole series of these probes specific for different caspases. So we have a probe for caspase 2, one for 3,7, and a polycaspase, meaning that it's essentially it's supposed to be specific to the whole family. And so what we actually found is that while over time we saw activation of specific probes, it was really this poly probe or this broad specificity caspase that really started signaling at 9 hours. So then we became interested, well, what is it really reacting with in this case? And what is this, activation of caspases doing? So there's kind of two ideas that could be going on here. Is this caspase activation required for viral replication? Meaning, is the virus turning on apoptosis in the cell? So why would a virus want a cell to commit suicide? Well, one reason is when a cell dies by apoptosis, it essentially dismembers itself without an immune response being triggered. Well, that's exactly what a virus wants, right? It wants to be able to replicate without the body becoming aware that it's in there. So a lot of viruses use apoptosis. The other reason is when a cell dies by apoptosis, this is a very programmed process. It takes about 24 hours from initiation to the cell finally being dissolved. So again, that gives the virus plenty of time to replicate. So that's a good reason that the virus could be triggering apoptosis. But cells also undergo apoptosis when they know they're infected. So we don't know exactly what's going on. But another question is how does the caspase activation affect cell survival? So to look at this, we did an experiment where we have our cells, and we're going to add this caspase probe, which, if you think about it, it's really an inhibitor. So because once that enzyme has been tagged with our probe, it's no longer active. So we thought what we'll do is we will add these probes ahead of time. And so what we do, so you can see here is, so here are the cells control. So no virus, no inhibitor, or no probe. Here are the cells with the inhibitor, and what we can see is essentially if we add these caspase probes, they don't do anything to cells if they haven't been infected with viruses. Now, in the case where, so in the top those are cells that are just virally infected, no inhibitor, and this is in the presence of virus and inhibitor. And what we see is by this time point, almost all of these cells have started to ball up and die. So much more quickly than with the virus alone. In fact, if we did a cell viability count, what we saw is, so the control cells, essentially 100% are alive. The inhibitor doesn't kill the cells. The virus, at the end of this actually is we're at 18 hours here. The virus has started to kill the cells. But if we add the inhibitor and there's infection, the cells die really quickly. Okay? So it appears in this case that actually this apoptotic process is keeping the cells alive. So it may be to the virus's advantage to start apoptosis. And actually, you actually get less virus out by the reduced plaque-forming units when you recover your virus. So we want to know what is the actual protein that's being activated here. And so we kind of switched this around. Instead of using a fluorescent probe, we used a probe with a biotin tag. So we could throw our probe in, react it with the cells during infection. We lyse the cells. We pass them over streptavidin beads. We essentially take those beads, we add trypsin, digest the proteins right off the beads. We put those into a mass spec, and boom, out pops a protein. Well, the protein that popped out is actually cathepsin. Well, this is not a caspase. Okay, so the way the manufacturers tell you and in the literature, this probe should not be recognizing this enzyme, but it clearly seems to be. But does that make sense? Well, yes, cathepsin is a cysteine protease, so it has a very similar active site. Also, it's in perfect position for apoptosis. So cathepsin, actually, if you remember, is one of the first things that gets activated when a cell senses its signal. It recognizes apoptotic signals. So this is actually even upstream of the mitochondria. So this occurs very earlier, far earlier than caspase activation, which shouldn't occur until 6 or 8 hours at the earliest after you trigger apoptosis. So this made sense kind of from our analysis that we saw things happening very early, and sure enough, we went back and redid our live cell imaging, and even 2 hours after infection, we can see this activation of these caspases. So it appears that this is a really early signal that's being turned on, very soon after the virus contacts the cell. It somehow is initiating activation of cathepsins. And so just kind of a summary is the majority of the regulated proteins that we found were associated with apoptosis. That was kind of our trigger that, hey, apoptosis is important here. This cathepsin becomes activated very soon after infection. And then by coupling, the proteomics with this activity-based protein profiling, we really came up with an unexpected mechanism, because this really wasn't on the radar in the literature at all, that we would find this. And so this is one advantage of kind of a proteomics or an omics style experiment, is you can come to conclusions that you would likely have missed if you had to only, proceed by, hypothesis-driven research, at least at the initiation of a study. And then these inhibitors, while they're, widely used in the literature, I think people have to be really careful about what are you actually inhibiting when you add, a broad spectrum inhibitor or even a drug. We think drugs are very specific, but it's often because people did assays and they only looked for a few things to be turned on or off. They weren't looking very broadly in a lot of these studies. And so now we're trying to, think more along those lines. Okay, now I'd like to shift to another project we have going on, hemorrhagic shock. And this essentially we want warning signs of shock. So what is hemorrhagic shock? So this is a clinical syndrome resulting from decreased blood and oxygen perfusion of vital organs resulting from a loss of blood volume. This accounts for about half of the 150,000 deaths in the U.S. attributed to traumatic injury. And it's a priming event for multiple organ failure. And right now, there are no clinical markers that a doctor can look at, like in the emergency room, to say, oh, we have 2 patients, they both lost blood in an accident, let's say, which one is likely to go into shock and which one is not. The reason this is important is the treatments for progression into shock are very severe, and they actually are very dangerous. And so if a doctor does not have to do that, they would rather not. Because if the person doesn't go into shock, the best thing a doctor can do is nothing. Is to just stabilize the person and let them recover. And so this is why it would be nice if they had biomarkers for shock. So this is a project that we're actually doing in collaboration with a colonel in the U.S. Navy. And you can imagine that the armed forces are very interested in this also, from the idea of battlefield situations and having to do triage. But so the general idea we have is that during, that metabolic changes arise early in the process of hemorrhage that predispose the patient to the course of events leading to, these complications such as multiple organ failure. And so what we want to do is examine metabolic changes and we're going to use a clinically relevant large animal model that Colonel Greg Beelman has to look at this. And so he does NMR. We're focusing on mass spectrometry to do this. And so we're doing this mass spectrometry-based metabolomics, where essentially we take a sample, and this could be metabolites from plasma in blood. They could be a urine sample. It could be a muscle biopsy. And essentially we take all the small molecules, and in a very complex mixture, we throw them into a liquid chromatography mass spectrometry system. We typically separate these compounds over a 10 to a 1 hour long chromatography gradient, and so that's essentially these red peaks you're seeing here. And then essentially for every second during that run, we have a mass spectrum, and that's showing the black line coming out. So we have both retention time information and mass information on up to 10,000 compounds in a single run. We get huge amounts of data. So there's lots of advantages. This technique has really high dynamic range, like 4 orders of magnitude of linearity. It's highly sensitive. We're detecting down in the nano and picomole concentration ranges. It's very fast. We can collect data on 10,000 compounds in an hour. And we get lots of data. The disadvantage is it's not comprehensive. We aren't seeing everything. Even with these large numbers of molecules, compound identification is very difficult. What we see is a molecular feature. We don't know what that is, and that's a big issue. And a big disadvantage is lots of data. But I just told you that was an advantage. Well, it depends how you look at it. When you have data files that are gigabytes of data and you have dozens of these, just handling this much data becomes a real big issue. Moving files around is now no longer a drag and drop thing. And trying to compare data sets that are this big, again, is very problematic. So it becomes difficult to actually work with these amounts of data. So in this standard approach, as I told you, we get a molecular weight for a molecule. Now, luckily there are lots of databases around the country now that have sprouted up, and one is this Metlin database. So it has over 25,000 compounds in it. So we can essentially match a mass to a compound in that database. And if we get a hit, then it'll pop up on a list. And so we can essentially measure our mass data accurately enough to, in the most times, generate a chemical formula right from the accuracy of our mass. And so then sometimes that leads to a single compound. Often, though, one chemical formula, so shown here, these top 2 compounds, look, this is the same compound. These compounds down here, for example, these 3 have the same chemical formula. So we don't have any more information than that initially. If we want to determine beyond which 3 of these it is, then we have to do another set of experiments to do that. But let's start first with saying, okay, we can identify compounds. We can actually do this quantitatively between one sample and the next. So the first problem we ran into in this experiment with trying to find a biological marker for hemorrhagic shock was, well, probably a bad choice. We decided to use urine. Well, one, because urine is really easy. It's a very nice, it's a very clean liquid from an analytical standpoint. There's very few protein contaminations. It's only got small molecules. So in that sense, it's easy to work with. Our problem, though, you can see here is, so these are different animals. So we have our animals that are under stress and our animals that are control, and these are at different time points. We have 5 different time points through this experiment. And you can see these urine outputs are all over the place. The problem with urine is we're essentially looking at molecules in liquid. And so if we have huge difference in outputs, we're going to have huge difference in concentration between samples. And so if you're trying to make a quantitative comparison, that's a problem. What we also saw is that if we combine our animals and just look at over time, we have big differences in that occur over time. So now we know we're going to have a lot of variability in our samples. Second problem. So now we're looking, so what this is showing you is a liquid chromatography chromatogram from a reverse phase chromatography. So this is time along the x-axis here, and this is intensity along the y-axis here. And so you don't need to be a mass spectrometrist to know that, wow, the red one and the blue one look really different. So essentially this intensity is just the ions. And so we can see peaks of ions coming out, but you can see that the red and blue look very different. The problem is these are two control animals, okay? This is just biological variability. And you can't get much more similar than these. These are pigs that have been fed on the same diet for months. They eat the same amount every day. They can drink all they want, but everything else is regulated. And look at how different their samples are. So then we were like, oh no, what have we gotten ourselves into? This is not good. If we look kind of a little more carefully at this biological variation, what we saw is that a number of the features can be different between samples. Some are essentially very similar, just slight changes in concentration, or they can even be exactly the same. So it's not a consistent change. So for example, the difference we saw was not just due to dilution, that one animal had a larger urine output than the other. Now you might just be saying, well, that's just because your mass spectrometer sucks. It's totally all over the place, right? Well, it turns out it's not, with very little technical variation. And so that's what it shows you. So this is 3 runs that are overlaid of one sample. And if we even go in on a close-up, you essentially cannot tell the difference between these. So our technical variability is very low. It's just our biological variability is really high in these experiments. But at least we know what we collect, we should be able to believe. So we wanted to check, though, if we had samples where it was very concentrated and very dilute, can we still quantitate between those? Because if we can, then this allows us to maybe do some sort of transformation or correction for urine output to adjust our samples. And so this just shows you we made actually a series of dilutions of a mixture of urine samples, and we injected different amounts. And then these standard curves here just show you we picked out some selected features and show that, we actually can, quantitate on these. We can get a nice standard curve. That the signal we see really does relate to the amount of material, whether it's from a concentrated sample or a dilute sample. So now we did our experiment, and we're analyzing our 4 control animals and 4 animals that undergo shock. They all start out the same at time 0. And so what I'm showing you here are principal component analyses. So this is a statistical test that really, what it's used for primarily is to tell you, do your samples look the same or do they look different? Essentially, it's a nice test for that. We're looking at what are the principal differences between a sample. And so we have our different time points, time 0, 45 minutes, 2 hours, 8 hours, 20 hours, and 48 hours. So time 0, they're all the same. They're actually not in any particular group at that time. By 48 hours, the animals have all recovered. They all appear to be healthy again. So what our initial analysis, this tells us is you can see that, so the red is the shock animals, the blue is the control. There is really no separation here. So we'd like to see the red spots in one area and the blue spots in another to tell us our samples are really different. What we see is, for example, in T0, T3/4, they're all over the place. At T2, so 2 hours, at kind of the height of this shock, there is sort of a separation. You can see in the vertical axis, we can actually separate control and shock, and then they become mixed again. So we don't have a lot of separation, but we have some. So then we actually played a few tricks with our data. and we actually did essentially a data transformation where we looked, instead of absolute changes between samples, we looked at median changes, and then we normalized that data based on the intensity. We converted it to log intensity instead of absolute intensity, and what we saw is now immediately, so at T0 we still can't tell the difference, but that's actually a good thing because there should be no difference in the animals at time 0. But we see now at 45 minutes, at 2 hours, we have a big difference. This is what we want to see, where now those biological controls that were, the urine output was all over the place, the animals started out different. When they're in shock, we can they're really separated. That we can say there are features in these samples that are really different. We can still do that at 8 hours. At 20 hours, when the animals start to actually recover from this shock, they come back together, and by 48 hours, they're starting to overlap again. So now we have a method that actually, something in these samples, we don't know what it is yet, is allowing us essentially to tell the difference between these two. But this is a control and a sample, set that we essentially, used. So 4 animals control, 4 animals sample set. And so we get a large number of features, and this is essentially just the probabilities of these features to tell the difference between these. But the first question is, well, are these features present in different animals at different times? Because this is just one group of animals that all underwent this shock treatment at the same time. So the question is this going to be something that we could apply to another set of animals? And so what we did is we took our analytical set that I just showed you about, and we had another group of animals that were actually had the same treatment done 6 months apart, and we analyzed this data 6 months later. So there's no overlap at all. And we compared, and so along the top line, we're looking at essentially the overlap of molecular features between these two data sets. And what we see is that within the group, there actually still is a fair amount of overlap between the data you just saw and a completely unrelated data set on animals. So then we said, well, what about a single animal, though? Because that's the more realistic situation, right? Someone's going to appear at a hospital, and you're going to have to take a sample, and you want to know, from that one sample, can I tell if this person is likely to be in a shock, going into shock or not? And so we looked at one animal, and we saw that while the overlap wasn't quite as good, it was still pretty good, considering how variable these samples were. So then we did this same principal component analysis where we added in 4 new samples of animals that were into shock and control, and essentially, so now we have 8 circles in here, and essentially they perfectly overlap. So even though this work was done 4 or 6 months apart, we can still essentially perfectly partition the animals between either a control and a shock group. And so another way to do this is actually to use hierarchical clustering, which this allows us to actually put significant values on this. And what we can see is that our control samples all cluster here. Here's our first group of shock samples. Here's our second group of shock samples. And essentially the length of these branch points tell us our confidence. So we're completely confident in our separation between control and shock. Now you can see that these animals that were analyzed at a different time, we can separate those fairly strongly from the original group, but they're still very different from the control group in there. So now we have actually a fair amount of confidence that we really can tell the difference between these. But this is looking at global features. So what if we look at individual metabolites? Because what we really want is a specific feature or a couple of features or a fingerprint that we can use to follow shock. And so now if we're looking at our analytical set, out of all of the features we detected, so there were 3,000, or almost 3,000 between the animals we were looking at, we narrowed that down to 247, which had the best predictive power. And so on this plot, there are 247 spots. And so now we're doing a principal component analysis, just looking at those 247 spots from each of the control and the stress group. And we can see, so essentially any pair of these spots that we pick can help us separate these animals because they're in a different position. If we go to a single animal, so that case, which is what may actually show up in your emergency room, we can see that not all the features have the same ability to separate, but you can imagine that if we pick 3 or 4 of these features, we're actually going to have a very robust test potentially to separate shock from control in this case. It's just a single sample on a single time point. So we're actually, okay, I didn't have a little summary here. So we're actually in the process right now. We're applying for one of these IDEA grants, so one of these local, you know, the Montana, Washington, New Mexico, Idaho, Oregon grants that they're involved with clinical translation. And so we're trying to get Bozeman Deaconess and a hospital in Billings involved with giving us some human samples to see, can we actually test a few human samples and see, can we find the same markers in these human samples? And then we potentially, I think, would have enough to really get an NIH grant on this to move forward with, you know, a fairly large, you know, trial type situation. Okay, and one last quick little story here is what everybody likes to hear. Yeah, that's really cool, but that's hard. Is there an easier way to do this? And so this is our idea. We're trying to actually think about looking at using a protein sensor assay. And so we're actually going to look at wine and urine, not together. So I want to start out with telling you about human serum albumin. So this is the most abundant carrier protein in our blood. So we have, our blood is filled with human serum albumin. This is a transport molecule. Its job is to move things around your body. So it moves a lot of endogenous molecules around our body, lipids, it moves signaling molecules, it moves hormones around our body. The other thing it moves around is drugs. So when you take a drug, oftentimes they're binding to serum albumin and being transported around the body. And so this has been really well studied because of that. And in fact, so if we look at the structure of human serum albumin, it actually has a number of different binding pockets, and they bind to specifically different drugs. So diazepam specifically binds to this 3A pocket. Okay, so each of these pockets has a little bit of different properties that they bind. And once drugs bind, there's actually kind of a coordinated effect, meaning this protein is allosterically regulated. Once one thing binds, it changes conformation, which can infect the binding of another molecule. Okay? Now, I guess about a year and a half ago, I was sitting in a seminar by Eric Anslin. So he actually came to MSU and gave a talk, and part of his talk was this kind of little fun part where he was saying he was going to, you know, he's using serum albumin essentially as a pattern recognition molecule, that he was showing he could detect different terpenes in perfume by essentially a fluorescent signal that he engineered to come out of serum albumin based on this allosteric response. But it was a very precise assay where he essentially ran standards, and then he showed that then if he followed, he could identify an unknown that was in his standards based on the fluorescent signal. Well, he also did this using wine. So he essentially looked at organic acids found in wine, and he showed that he could, using his BSA, he could specifically tell you if there was a type of organic acid in his wine. Well, we thought that was cool, but gosh, it's kind of limited because he had to know exactly what he was looking for before he started. And it turns out it wasn't very sensitive because he was using a fluorescence assay. So we said, well, what if you just use the protein sensor and a mass spec? And so we came up with this idea of a simple assay. What if we just took BSA, we throw it into a complex biological solution, urine or wine, for example. We let the molecules bind to the BSA, we wash those off, and we take what's really stuck on there, and we essentially elute that, and we throw those into our mass spec and say, hey, can we tell the difference? So, you know, as the first quick experiment, my graduate student ran down to the store and bought a bottle of Cabernet Sauvignon, Merlot, and Pinot Noir. We're going to start with red wines. Not sure why we started with reds, but we did start with reds. And so he repeated this assay 3 times, and so I'm showing you these principal component plots again. So we're looking at 3 different components, essentially, to look at the variability. And again, this is just to show you, is our data reproducible? Do we have differences in our sample? And I think, as you can see, the individual samples cluster really tightly, and they're very separated from each other. So 30 minutes after opening a bottle of wine, we can tell you, if we have 3 choices, we can tell you, is it a Cab, is it a Merlot, is it a Pinot? So we said, well, what about when the wine breathes, right? You're supposed to let your wine breathe. Okay, we'll be a little extensive and we'll wait a whole day. There was wine left by the next day. We tried this at 24 hours. Wow, we can tell the difference at 24 hours, but it was a little bit different. Things had changed, and we actually mix the 2 analyses together. What we see is we can tell the difference in the varietals, and we can still tell the difference in the time, although the time separation is much less than the difference between the varietals. Okay? So then we're kind of happy, wow, the assay, I think this might work. So, of course, the next thing is white wine, right? If you can do red, can you do white? And so now we ask a little more challenging question. We said, well, it's increased the number of varietals, but let's get some of the same varietal from different vineyards. And so we got a couple of Sauvignon Blancs, we got a couple of Chardonnays, and we actually got 2 blends, one that was primarily a Grenache, and then another one that was a Voyeur Chardonnay blend. So we did our little binding assay, we threw the samples in the mass spec, and we ran them. And so again, there's our principal component analysis up there showing you that they partition, and then here's showing you a hierarchical clustering, where again we can see which cluster together. And so the first you can see is that the technical replicates are all right together. So the method is reproducible. But then what was nice to see is actually the varietals cluster together, but they're clearly separate. So we can tell the difference between the same wine from different vineyards, but also we can then tell that wine from a different set of wines. So we've actually now got a more complicated one. We've actually done 29 different red wines. I was forced to go to a wine tasting and collect samples to do that, but we can do the same thing with a very complex set of wines and essentially tell cluster varietals in almost every case, and then but tell the difference between which vineyard they were actually at. So one of the questions, though, is we're just saying we can tell them apart. We're not telling you why. We didn't know why we could tell them apart. And so then we wanted to go in and what are the small molecules that are causing this separation? So we went in and did an analysis essentially looking for what those molecules were, and we had a surprising finding. One of the things that immediately popped out was a number of pesticides that we were finding in these wines. Now these aren't complete identifications. These are tentative identifications. We've matched them to a database, and they make sense, but it doesn't mean that we've identified all these pesticides in these wines. But another interesting point was we then ran the wine samples before putting them over our protein sensor assay. The number of the number of pesticides we could detect was actually lower. So it appears that by using this protein sensor, we're actually enhancing our ability to detect these pesticides, which suggests that they may be at very low concentrations. But remember back to what BSA does. This is I told you about human serum albumin. We're actually using the cow version in our experiments. What does this protein do? It binds to small molecules and carries them around. So it appears that it is preferentially binding to these pesticide-like molecules. And we actually have an expanded list in this protein assay. So we're actually in the process now of, this is something that's of interest because we're trying to then confirm these pesticides presence, because we've actually done a number of organic wines. And we find a smaller list, but a fair number of pesticides in the organic wines as well. Now, to the end. Back to our hemorrhagic shock samples. So remember I told you it was difficult for us to separate the control from the shock if we didn't do this. It's a fairly sophisticated data analysis, this transformation that we do. What about BSA? Can we use this protein sensor assay? And so here is a principal component analysis showing you our different time points for our hemorrhagic shock samples. I just want you to focus on the squares. This is time 0. Shock and control, they're right on top of each other. Before the experiment starts, we can't tell the animals apart. When we get to our clinically relevant 2-hour time point, the spheres, now in this assay, our samples are completely separated again. Okay? So even in these urine samples, this protein sensor assay appears to be working, allowing us to separate. The really neat thing is we can do these chromatography runs much more quickly because this reduces the complexity. So now we have a way to speed up our analyses. It doesn't take half an hour or 45 minutes. We can now do this in 5 and 10 minutes, which again would be very important in the assay. So then with the shock samples, we ask again, well, what are the molecules that are important? And so the top of our list for being most regulated between the shock and the control, here's our the list of the top 8. Creatinine was at the top of our list. Now, this is a molecule we completely expect to find in urine. It should be in your urine, but it's actually a marker for metabolic stress in your body, although no one had connected it directly with shock before. So we went in to prove then, are we really seeing creatinine? Is it not just a database hit? So we actually purchased this molecule. What we did is so this is showing you chromatograms where we take our sample, and that's shown in the green peak, and then we actually spike creatinine into a urine sample, and we see where it comes out. And so we see that they coalute, they're within a second or two of each other. And then we look at the mass spectrum, and what we do is we actually don't only look at the intact protein mass, but we do MSMS. We fragment this molecule at different levels of energy, and we see that our fragmentation patterns are exactly the same within the sample, our standard, and then our spiked sample. And so this is confirmation that that is the molecule that we're seeing. So now we can say we have an identification of creatinine in these samples. So now again, we're moving toward seeing if we can develop this, because if we have one specific molecule, now we can think about designing a very rapid test for looking for hemorrhagic shock. So this question, does the protein sensor work? Well, I didn't show you this example, but this is a set of oxidative stress samples where if we don't pre-treat our samples, essentially they're all over the place. We can't tell the difference between different time points of stress. Once we treat with our protein sensor, we can immediately separate these samples. I showed you this wine work where this is our red wine work where we can still, so this is actually samples that were not treated with the protein sensor assay. We can still separate some, but our separation gets better. And the most dramatic example is our hemorrhagic shock. If we don't use the protein sensor, essentially the samples are all over. We can't tell the difference. As soon as we use the protein sensor, we can really differentiate these two samples and tell the difference. So we think that this actually does have some potential utility, and that's because, one, it's fast and reproducible. We can decrease our runtime by 50%, and actually since I made this slide, we're actually down another 50% or more. And it works really well with, remember, these urine samples. We're not adjusting for concentration. So the fact of this protein sensor is kind of normalizing these samples and concentrating, right? We're pulling these things like these pesticides that a number of them were below our detection level before we added them to this assay. Now they're up where we can detect them in our samples. And we can discriminate from these complex biological systems. So I'd just like to point out to you guys that these are the people who really did the work, and I have a wonderful group of people in my lab, and I should say that the whole department at MSU, so any of you students or you professors who have students who they're thinking about graduate school, we have a really phenomenal department doing lots of different things. So send students our way. We love it. And there's my greater Bothner group. And I'd like to thank my collaborators on these projects, and of course our wonderful funding agencies that I hear actually are open now, and the government is back on. Cool, we got to applaud that too. And thank you all for your attention.
Does anybody have any questions? We'll do a minute or two of questions. I know that it's after 5, so if any of you guys need to